ISMB organizer for allowing me and my collaborators to present our work. So this work has been dedicated to the production of a new reconciliation approach to reconstruct the evolutionary history of gene synthesis using a duplication, transfer, and loss model. And I will present you an application of this model for the study of the evolution of Cas gene synthesis. So first, I need to explain what is a gene synthesis. So a gene synthesis is a set of closely located genes that can be at once affected by segmental events, such as segmental loss, duplication, or transfer. Here is an example of a synthesis where we have 4 genes A, B, C, D that are compose the synthesis. And we have an example of segmental duplication. We have the segment composed of the 3 gene B, C, D that is duplicated. So the copy is passed on another part of the genome, and we have still the initial copy of the synthesis. So here in this study, we are interested in the problem we addressed is given a family of homologous syntheses, infer a plausible scenario for the history made up of segmental events. So first, also, I would like to present you the gene syntheses we are interested in, that are the Cas gene syntheses. Our Cas gene syntheses are part of the CRISPR-Cas system that is now a very well-known system because it's used in gene editing technology. So initially, it's a mechanism used by prokaryotes. It's a human mechanism that allows the prokaryotes to resist against the bacteriophages invasion. So the CRISPR-Cas system is composed in 3 parts: the Cas gene synthesis, a leader sequence, and the CRISPR array. So the CRISPR, it's for clustered regularly interspace short palindromic repeats. So between the repeats, so we have a space. It's a spacer sequence that corresponds to the target sequence the mechanism wants to recognize and to remove from the cell. So to make most of the function of the CRISPR-Cas system, most of the functions are given by the Cas gene. There are some of Cas proteins that are involved in the adaptation that consists to integrate the target sequence into the CRISPR array. There are Cas proteins that are involved in the transcription of the CRISPR array as a CRISPR RNA, so it will be recruited at the level of the leader sequence, and it will transcribe the CRISPR array. And after, you have other Cas proteins that are involved in the interference that will be guided by the CRISPR RNA and allow to cut and remove the invasive sequence from the cell. So here, to reconstruct the Cas gene synthesis history, we wanted to present you a super reconciliation. So the super reconciliation is an extension of the gene tree reconciliation method, but to infer the history of gene synthesis. The context is taken as a problem input. It's a synthesis tree that will replace the gene tree. And after, we will fit the synthesis tree into the species tree to reconcile, to infer the evolutionary history of a synthesis. So we call it a super reconciliation due to the fact that synthesis tree can be obtained as a super tree of individual genes. And it was first used in the publication from Matteo and colleagues in 2020. So to model the synthesis, there is 2 possible ways. We can consider a synthesis as an ordered synthesis or unordered synthesis. In the ordered synthesis, you have a set of genes that are ordered, but in the unordered, the genes are unordered. So in our method, we model the synthesis as unordered set of genes because it doesn't require to be order consistent synthesis, because if you want to take in account the order of genes, the gene order have to be consistent between synthesis, or you have to model the rearrangement process inside the synthesis to model the evolution. But it's particularly computationally extensive, so we make the choice to use unordered set of genes. After that, we need also to model the segmental event. So we are in a duplication, transfer, and loss model. And segmental events can be divided in 2 categories. The binary event, where you have the production of a new synthesis, a unary event, or where you have an existing synteny that will be modified. So in the binary event, you have first the speciation where you have an ancestral synteny after splitting of the ancestral splitting in two species gives two new synthenies. The duplication, so decretion from a complete or part of the synteny giving two new synthenies. And after the transfer, you have a duplication of part or complete of the synteny, but this copy is after transfer in another species. After you also the unary event, so you have the loss event where you have a part of the synteny that is lost, but you still have only one synteny. And you have also the gain, so the synteny gain genes. So in our model, the gain are non-segmental. We only gain gene once, and each gene, so gene family, is gained exactly one because we are based on the parsimonious model. So it doesn't allow to have convergent evolution. So as I said, SuperDTL model reconciliation is a parsimony problem. So the idea is to find the most plausible history for a given synteny and species tree. Each event is given a cost value, quantifying its rareness, and the cost of the history is the sum of cost for each event. So our problem is a minimum cost history problem that take as input a synteny tree T, a species tree S, and this will output a minimum cost history corresponding to T that will fit into S. So here is a slight description of the algorithm as we take as input a synteny tree and a species tree. So the first step will consist to map the synteny tree into the species tree. So here is a step where you have to fit into first step of reconciliation. After you have the assignment of ancestral synthenies. And finally, you have the assignment of evolutionary event along the evolutionary history of synteny. So I won't enter in the detail of the algorithm because I want to present you the result we obtain on the CRISPR-Cas gene synteny. So here, to study the evolution of Cas gene synteny, we were based on the dataset of 15 bacterial Cas synteny from Makarva and al. So this set is composed only of Cas gene synteny from the class 1, and from our dataset, we remove the gene repetitions. After, we take also as input the synteny tree topology from the same article, where there is one synteny per species and one species per subtype, but also this synteny tree includes multiplication. And finally, as input, we take also a species tree based on the topology from Coleman and al. That is the last up-to-date phylogeny of bacterial species. So here is the dataset. Here on the left, you can see the species trees. On the right, the synteny trees, where we have here the representation for each synteny, which Cas genes are present in each synteny. And on the right, the topology of the synteny tree with, in color, the different types of the synteny tree. And as we can see, there is it corresponds the middle corresponds to the correspondence between the species tree and the synteny. And we see there is a lot of inconsistency between the species tree and synteny tree that can say that there is some transfer between the bacterial species. So the need to consider transfer to reconstruct this evolutionary history. So here, as I say, we have a multifaceted Cas synteny tree. So as input, we use all resolution of the synteny tree. And we test for all of this synteny tree resolution different evolutionary costs. So we put the cost of the loss to 1, and we try different cost for duplication and transfer. And here is a table representing the number of solution parsimonious solutions that have been recovered for the different parameters. On the column, you have the cost for duplication, and on the line, the cost for transfer. And we see that it's for cost of transfer for 4 and several costs for duplication. We have the lowest number of solution, and we analyze in detail this result, and we see not a lot of difference. Most of them is due to sometimes the position of a transfer when you have a speciation followed by the loss. Sometimes the transfer is before or after this node. So we, beyond the 32 solution, we take one of the solutions that seems for us the most plausible evolutionary history. So here is the evolutionary history of the CRISPR-Cas gene synteny of class 1. So we see there is the emergence of the Cas synteny at the root of the Terra bacteria species. So we have the Cas gene 5, 6, 7, 8, 10, and 11. We after see the emergence of the type 1 and type 3 at the root in the ancestor of the cyanobacteria after the inclusion of the Cas gene 1 and 2. After at the level of the ancestor of cyanobacteria, we see that for type 1 there was the inclusion of a Cas3 gene that is leading to the new type 1. And when we see this evolutionary history, it's mostly in line with the evolutionary scenario from Kunin and Makarova. That is the last up-to-date hypothetical evolutionary history of CRISPR-Cas synteny evolution, except to the position of the type 1,4. It is more in the model. So the supposition is more that type 4 it's a degraded ancient CRISPR-Cas, but in our posthumous model we consider more the type 4 as an ancestral state, and after you have the derivation of the type 1 and type 3. And as we can see on this part of the tree, we infer history with a duplication followed with several loss here, leading to the synteny in Escherichia coli. So this history is a little bit time cost consuming. And a more plausible history would be there is a transfer on the unsupple species, and after they simply come back in the species tree at the level of the Escherichia coli. But for now, in our model, we don't consider the possibility to consider unsupple or extant species. So here is one of our future work we would like to do. So consider unsupple species to act as intermediate in the transfer of synteny. We'd like also to improve our dataset by using the database CRISPR-Cas++ that contains a lot of information about Cas synteny. For example, in our study, we don't include archaeal species. There was only bacterial, but we know that it's about 40% of bacteria that possess a CRISPR-Cas system, while in the archaea it's more about 70% of species. So it will be interesting to consider archaeal species to reconstruct the evolutionary of Cas gene synteny. Also, in this study, in the paper from Makarova, there was only one gene synteny per species, but we know from this database that some species are more than one Cas gene synteny. So we'd like to produce a more complete data set to fully understand or more generally understand the evolution of Cas gene synteny. And also, we have the need to develop methods to infer more accurate synteny trees. For now, we take an input synteny tree, but there is a very important need to have accurate methods to infer synteny trees to infer correct evolution history. Also, we have some complexity improvement to do on our work. So thank you. I would like to thank my co-authors, Matteo Delabre and Nadia Helmabrook. I would like also to thank my lab mates like Mathieu Gascon that will present another talk this afternoon on non-binary tree reconciliation with endosymbiotic gene transfer, and also my teammate in the University of Sherbrooke, my supervisor Aida Wangra, Ibrahim Shikran that is presenting a poster on model selection for non-coding RNA classification. Abby Devi that will present tomorrow, a database on the transcript to study the transcript conservation and evolution with Gen3. And that's it. Thank you for your listening. And their work have been published in recently at the last conference of EvolCompGen. And if you want more about this.